Welcome everyone to the second part of the webinar. In the next 20 minutes or so, I'm going to guide you through how high-throughput transcriptomics can be used for early drug discovery and specifically for compound screening and lead optimization. Here's the agenda for today. So I would like to tell you about how Alpern's drug-seq technology accelerates drug discovery. I will showcase a couple of examples of the powerful combination between drug-seq and AVITI instruments for rapid compound profiling. And then I also would like to take a bit of time to introduce you a new total drug-seq technology that enables more deep mechanistic insights. So as everyone knows, drug discovery is a long, costly, and very high-risk process. It often takes decades and billions of dollars to develop molecules, but yet 90% of drug candidates fail in clinical trials. So there are multiple reasons for that, but the major reason is that toxicity is something that was not detectable in early stage drug development, and that only becomes evident in the late clinical phase, which makes this failure actually very costly and very unfortunate, for the field and also for the patients. So the consensus in the field again is that there's a need for deeper understanding of biology to be able to prevent or predict the toxicity early on. And for that, there's a couple of bold strategies that are being now employed by the industry, including usage of more advanced experimental models that will be more physiologically relevant and genetically relevant for humans and specifically for patients. This includes 3D cultures and patient-derived samples, such as iPS and organoids. Multi-omics enables a deep mechanistic dive, basically, revealing complex phenotypes. So something that was just showcased by Wilfried in the previous talk. So multi-omics typically results in very sizable data sets that basically requires the machine learning models and AI integration for improved data-driven discovery. So these three pillars are believed to be the way to reduce the failures in drug discovery and on compound discovery, actually, not only in the pharmaceutical industry, but also other chemical industries such as agrochemistry and cosmetics. So multi-omics is basically the integration of diverse molecular data layers that often includes transcriptomics, phenomics, proteomics, metabolomics, and so on. And it offers altogether a comprehensive system-level understanding of biology, of disease or a compound effect, and the mechanism. And so, while all the layers are providing very highly complex molecular data, not all of them are compatible with the need for high throughput. And this need comes from the fact that when we screen compounds, we often rely on large libraries of chemical molecules that need to be tested. They need to pass through this design and test cycle multiple times in order to find the best leads. So thus far, there are only two omics that are actually bona fide high throughput. And this is phenomics, illustrated by the method that is called cell painting, that is already heavily employed by multiple labs for morphological profiling of cells upon perturbation. And the second omics would be transcriptomics. That is again really high throughput, and this is enabled by, in particular, drug-seq technology that is offered by Alpern. So Alpern was founded in 2020 with a mission to enable high-content molecular profiling for biomarker discovery and drug discovery. And since then, we offer kits that can be employed for transforming samples to the libraries. We offer bulk RNA sequencing kits and also single cell that was recently released. Flash-seq, we believe, is one of the most sensitive on the market. And besides kits, we also offer services in which our team is actually employing the kits to transform samples into the data. And on top of that, we also offer a high-throughput transcriptomic screening services that basically encompass the experimental planning, and in the partnership we can provide cell culture and perturbation. We can also provide complementary data with phenomics that we believe create also a powerful combination for more insights in the data. Also, something I wanted to mention is that Alitae recently received the ISO certification that covers both manufacturing and the service activities that we do. So what exactly make Alitae's MERCURIUS DRUG-seq ideal for high-throughput molecular profiling? Well, this method enables evaluation of the entire transcriptome in an unbiased manner, so we can detect genes by 3' counting, and this is focused on polyadenylated transcripts. It's very rapid sample-to-data transformation because it employs extraction-free procedure in which we can employ the lysate in the library preparation. Thanks to its early multiplexing technology, it's ultra-scalable, meaning that we could transform 96 to 384 samples in one library in a very quick protocol. And importantly, for high throughput, it's also cost efficient. So just to give you a little more kind of insight about the actual workflow, it starts with a frozen plate with the cells that is compatible basically with any format, 96, 384, and we're going to soon release a 1536 format highlighting Alitae's strive to deliver very high throughput library preparation. So this plate is used for dispensing the lysis buffer with the synthetic RNA that is used as a control. And after that, the lysates are transferred into the RT plate containing barcoded oligo-DTs. So each well is tagged with a sample-specific barcode that basically enables to pull all the samples in a single tube after the RT step. And after that, the library prep becomes a very straightforward process that would take a few more hours before loading that into the sequencer. The drug-seq libraries from Alitae are compatible with any type of the sequencing instrument currently available on the market. But we in particular like using the AVITIs because of multiple reasons that I'm going to talk about. But importantly, they're very fast and flexible. So after the sequencing data is obtained, we can bioinformatically, using standard workflows, demultiplex and obtain information for every sample. Just to illustrate to you, how this works in practice, I would like to tell you about a small project that we are working on together with our partner, Axon, which is a life science CRO in Italy. So in this project, we would like to profile 58 compounds with a known toxicity effect on liver and evaluate the response using transcriptomics. So we have 58 compounds in 8 doses and 3 replicates, which results in around 2,000 samples that were prepared in 9 384 plates, and it's just a part of this project because we will employ different cell lines. And so here it's just one cell line, and basically when we receive the plates, we prepared drug-seq libraries, and just one person can easily prepare that in 3 days. And those libraries were pulled together and subjected to AVITI sequencing. In a run that lasts just 22 hours, we could obtain enough sequencing reads per sample. So we require around 1 million reads per sample. And so when we obtain the data, as usual, we observe it's a high-quality data. And so, in a few slides, I would like to illustrate what happens next. When we get the data, obviously, the data that we obtain for drug-seq is pretty much similar to a typical single-cell data with a lot of data points. And given that there was a batching, it requires basically batch correction. And you can see that upon batch correction, there's a nice distribution of samples coming from different libraries, and you can see that rather than clustering by the technical batch, they would cluster by compound and dose. And you can see that some compounds render stronger effect, making some clusters to outline further. And there's a clear dose dependence for some of the compounds. So with the transcriptomics data, we could identify so-called points of departure, which would be the minimal doses that render a noticeable effect on the cell model. Here you can see the curves that illustrate the number of GE genes that are identified at increasing doses, and you can see that different compounds act differently. Some of them are just constantly increasing the number of GE genes, while others increasing and then going down, which is an indication of the toxicity. And so, providing just a few more examples, we can look at the gene signatures. Transcriptomics data is highly actionable with this regard because it provides information on many genes, and we could infer the mechanism of action by looking at the pathways. And moreover, we can also look at the similarity between different compounds. In here, a small example, you can see that two compounds are clustering together here, revealing the similar gene signature. And these compounds are actually known chemotherapy drugs, daunorubicin and doxorubicin, with the same mechanism of action, namely they are inhibitor of topoisomerase and DNA damaging agents. And this is why you can see that in the pathway enrichment, they demonstrate very similar signature involving p53 signaling pathway activation. And just to say that this signature is quite distant from the signature of another apoptosis-inducing agent. So we can clearly kind of identify the dissimilarity and similarity in the mode of action of different compounds using transcriptomics data. And what is more is that we could also, with the drug-seq data, we could evaluate the cell viability. So in Alitaeus technology, we employ spiking RNA that we add in every well as a control, but also this enables to evaluate the ratio between the host RNA, the RNA coming from the cells, and the spiking RNA. And by doing so, we can infer the cell viability. So if the cells are dying in the well, we could see that more reads will be assigned to the control RNA. And we developed a machine-based model for inferring the cell viability, and this is just a demonstration of how it works. So we can see a cluster here that displays a lower cell viability score. So there's a cell death observed in this sample, which is also confirmed by the pathway analysis of the samples here, which basically highlights that apoptotic pathways are being activated here. And this cluster also belongs to the cluster of the higher dose treatment, so which actually makes sense. So basically, the cells are undergoing apoptosis here because of the compound type and a high concentration. In contrast, you may have, you can have another cluster with also high dose, but in this case, no apoptosis and normal cell viability. So by doing that, we could also define between technical variation and actual perturbation-specific cell viability variation. Just to give you also a better idea of how AVITI is facilitating the drug-seq projects that we're running at Alitaea. So the instrument provides the perfect fit for small to medium-sized drug-seq projects. We really like it because it enables very fast data generation. It's very high quality, and it's also cost-effective. So, flexibility combined with accuracy and low cost really makes it the best instrument on the market for this purpose. We are using a very straightforward adapt workflow for library circularization. Team loves the instrument because it's very easy to use and very easy to maintain. And whenever we have a question, there's an amazing support team. Just to give you a few numbers, with one AVITI, we could obtain 2 billion reads a day using the both sites, which refers to capacity to profile around 2,000 drug-seq samples. And right now, as we speak, literally, our AVITIs are being upgraded to AVITI 24, which actually will boost the sequencing yield from the instruments 50%. So we expect that this just simple upgrade will enable to profile even more samples. And for our projects, we actually use 2 instruments that again enable us even higher flexibility. And maybe to illustrate that more scalable projects are compatible with AVITI instruments, here there's another example of a study that we are also running in collaboration with Axon, but this time we do it together for a customer. And the goal of this project is to characterize new compounds in different cell contexts. So we use 6 cell lines from different tissues to profile these compounds. And so in this project, we have, so it's a multi-phase, a multi-omics project for which we already completed a few phases, and in particular, one phase consisted of around 23,000 samples across 6 lines, as I mentioned. And so you can see here that we can detect in between 13 to 15,000 genes per sample, while processing each individual cell line in a batch. And just also to give you a little snapshot of the data, we could bypass all the technical variations by performing the batch correction, and the data is nicely integrated and reflects the dose, compound, and cell-specific response upon the treatment. And also just a little illustration here, you can observe some compounds are rendering a very clear cell-specific effect, so they're acting very strong on some cell lines, but not so much on other cell lines. Again, just illustrating the complexity of the biological systems and the need to use the right cell model to address the right questions. All right, now I'm shifting a little bit the gears, and I would also like to give you a little overview of the new technology that we also just released a few weeks ago. We call it total drug-seq, and it's basically features the key elements of standard drug-seq, but it enables to cover the full transcript length. And by doing so, we gain the capacity to identify different splicing isoforms. We can also identify genetic variants, transcription start site, and transcription stop sites. So the technology is based on polyethylation, so fragmentation and polyethylation of RNA molecules. And basically, after this step is done, we employ the barcoded oligonucleotides. But given that fragmentation is happening early on, this protocol is even quicker than typical drug-seq, still allowing for similar scalability. So as I mentioned, this is just an illustration of the fact that a total drug-seq covers the entire transcriptome and is comparable to the standard transcriptomics kits on the market, but on top of that, offering early multiplexing capacity. So in terms of the gene detection, the gene detection is pretty much similar between total drug-seq and 3' drug-seq. However, given again the fact that it's covering the entire length of the transcript, we substantially improve the detection of splicing isoforms. And this detection is even further improved by employing paired-end sequencing. And here I need to dive a little bit into technical detail, because paired-end sequencing with drug-seq libraries is not given. So there's a technical limitation, namely poly-T homopolymer sequence that is an inherent part of the library that makes it complicated to sequence these parts with the standard Illumina sequencing. However, for AVITI, this is not an issue. So there was a paper already some time ago demonstrating that elements AVITI could easily sequence through poly-T homopolymer sequence without losing the accuracy. And basically what we observe is the same. And by doing so, we could also recover the fragment coming from the read one and improve the mapping of specifically of the splice junctions. This is the key basically to detecting more splicing isoforms. And to illustrate how these data can be useful, this is a project that we run with liver carcinoma cells that were treated with TGF-beta, and this is a model for tumor progression. So TGF-beta induces epithelial mesenchymal transition in these cells, which is typically highlighted by the promoter usage shifts in HNF4 alpha transcription factor. So in the normal liver cells, the P1 constitutive promoter is active, resulting in the isoforms, in the normal isoforms of HNF4. But in early stages of cancer progression, or in MET, the promoter is shifted to P2, and that's what you can observe here. In standard drug-seq data, these would be missed, but in a full coverage, we could see that there's a gradual decrease in P1 activity for an increasing dose of TGF-beta, while there's an increase in P2 activation that happens simultaneously. So basically, at some point, they shift completely. And it's just an illustration, again, for how these data can be used to obtain even more fine-grained transcriptomics insights on the models in a very high throughput manner. All right, so that was my last slide, and I jumped to the conclusions. So I hope I managed to convince you that high throughput transcriptomics enables rapid and information-rich sample profiling at scale. A kit and a service that we offer at Alitea is a practical and very scalable solution for data acquisition. And then, in combination with a AVITI system, drug-seq offers very quick and reliable and flexible data generation for cost-effective data acquisition. All right, thank you very much. I'm happy to take questions.